Ketogenesis is one way the liver can biochemically produce a molecule from fats that the brain can use for energy. Remember, brain cells are primarily reliant on glucose for energy and can't use fats. So instead, acetyl-CoA can be converted to ketones when the levels rise from fat degradation. You may have heard about ketogenic diets, which exploit the biochemical pathways of fat degradation in order to lose weight. In this lecture, we'll see how ketone bodies are made in ketogenesis, and many of these diets use the measurement of ketone bodies to demonstrate that fat is being broken down. There is, however, a side effect, and we'll see that the production of these molecules generates lots of protons, which can lead to ketoacidosis. The hormonal regulation of the various pathways that we've covered is going to be important, and we'll review their involvement in particular for type 1 diabetes. We'll also see how hypoglycemia and hyperglycemia arise in our bodies and the effects of the hormones and metabolic pathways. These are the supplemental readings with the link to the textbook provided on Quirkus. And we're going to finish up this one little step in this pathway outline, looking at the production of ketone bodies from acetyl-CoA. Remember, acetyl-CoA can be produced from carbs, fats, or proteins, but we'll see that this deviation to ketone bodies out of the normal progression through the citric acid cycle and oxidative phosphorylation usually occurs when the levels of glucose in the body drop. We've already seen how the breakdown of fatty acid tails generates acetyl-CoA in beta oxidation, and how a single fatty acid tail generates approximately 3 times the amount of energy than a glucose molecule. There are also amino acids that can be used to generate acetyl-CoA and energy if the amino acids are being used for energy instead of protein production. Remember, the breakdown of fats and proteins will occur when your body is in the catabolic state, so either epinephrine or glucagon will be signaling via their GPCR signaling pathways, and this will allow for catabolism to occur and generate energy. In order for acetyl-CoA to be used in the citric acid cycle, the cycle is dependent on the amount of oxaloacetate that's present, since this was the last molecule that's made in the cycle and a starting substrate. If we don't have a lot of oxaloacetate, the citric acid cycle can't efficiently run, and an excess of acetyl-CoA can instead be used to make ketone bodies in the liver as an alternate fuel source to glucose. So the liver really is the provider for the body. It supplies glucose from glycogen breakdown and gluconeogenesis, as well as ketone bodies made in ketogenesis from acetyl-CoA. I've just outlined a few of the places where amino acids come into the various pathways, and these would be considered non-essential amino acids if they can easily be made from some of the intermediate metabolites of the pathways we've looked at. Depending on where they enter into the pathways, amino acids can either be classified as glucogenic or ketogenic amino acids. The glucogenic amino acids can be used to make glucose. So for example, glutamate can be converted to alpha-ketoglutarate in the citric acid cycle, and this will go through the rest of the cycle to generate energy and oxaloacetate. That oxaloacetate can then be used to make glucose in gluconeogenesis. And the same goes for these other 3 amino acids that are listed. The ketogenic amino acids can be used to make acetyl-CoA, but remember, acetyl-CoA can't be converted back to pyruvate to make glucose. So instead, they can be used to make ketone bodies in the liver, which the brain and other cells can use for energy. Acetyl-CoA really is at the heart of metabolism. It can be made in the catabolic pathways from the breakdown of carbs, fats, and proteins, and then used for energy in the citric acid cycle. But if there's an excess of acetyl-CoA, it can participate in another pathway where the liver will make these ketone bodies. And they're not as big as fats and can be converted back to acetyl-CoA for energy. So the default pathway in metabolism really is to make energy. Acetyl-CoA from carbs, fats, or amino acids will be converted to citrate in the citric acid cycle and eventually produce energy and the waste product, carbon dioxide. Now, what happens if we have enough energy? Remember, the enzymes of the citric acid cycle that had large drops in free energy can be regulated and inhibited by the production of energy. So this will slow down the citric acid cycle so that citrate can be exported for fat synthesis. The problem, though, is that it is dependent on the presence of oxaloacetate and energy. If we don't have enough oxaloacetate, remember this can come from carbohydrates, the level of acetyl-CoA will build up and it'll spill over to produce ketone bodies in the liver. These can then leave the liver and be used by other cells to produce ATP. These are the three different ketone bodies that are made in the liver, and they all fall under this category of a ketone body. On the left, I'm just reminding you of the structure of acetyl-CoA, and you can kind of see how molecules of these can come together so that the acetyl functional groups can be used to form these molecules. Now, there's no energy in the form of ATP that's required. It's that thiol group in the acetyl-CoA that helps drive their formation. And it is a bit of a misnomer as only 2 out of the 3 molecules include ketone functional groups. Beta-hydroxybutyrate doesn't have a ketone, it only has a carboxylic acid, while acetoacetate and acetone both still have that ketone group from acetyl-CoA. Now these first 2 have carboxylic acid groups on them, so when they are made, they actually release a proton due to the low pKa of this functional group. And this can actually contribute to acidosis or a drop in pH if too many of these ketone bodies are made. These molecules are highly soluble, and they are the ones that will get converted back to acetyl-CoA in the tissues. This third ketone body, acetone, is actually an exhaled waste product. It's made when acetoacetate is decarboxylated, which we've seen many times as a very favorable reaction. Some people may say that it kind of has a fruity smell to it, and it might be detectable on your breath, kind of like the fruity smell of nail polish remover. And there are actually devices you can buy to measure either acetone production in your breath or these other 2 ketone bodies in your bloodstream. And people might buy these and use these machines to measure ketogenesis, or also known as ketosis, in their body to detect fat metabolism. So as the levels of glucose in your body drop, the amount of oxaloacetate produced from pyruvate drops, and ketogenesis occurs as the acetyl-CoA levels start to build up. These ketone bodies will be made in the mitochondria of your liver, primarily from fat and protein catabolism, and acetoacetate and beta-hydroxybutyrate are the 2 that can be converted back to acetyl-CoA for energy. Acetone is the decarboxylated waste product and has a characteristic fruity smell that can be detected when it's exhaled on your breath. And when you have too many of these ketone bodies made, acidosis can occur as they release protons upon their synthesis. We all will have low levels of ketone bodies being synthesized, say first thing in the morning when you wake up, but your body is designed to buffer these protons and get rid of them. When the ketone bodies enter other tissues, and remember your brain can't use those long fatty acid tails, but they can use the smaller ketone bodies for energy. Since they don't have those coenzyme A attached, they can cross membranes and the blood-brain barrier, and they're very soluble so they can travel easily in the bloodstream and then get converted back to acetyl-CoA in the cell. Beta-hydroxybutyrate will be oxidized back to acetoacetate, and that will also make an NADH that can enter into the electron transport chain. And then we'll have 2 coenzyme A's that'll be used to recreate the 2 acetyl-CoAs, and you can see how this pathway is linked to the citric acid cycle. We actually use succinyl-CoA as a CoA donor. Each of these acetyl-CoAs can then enter into the citric acid cycle to make ATP. Now, don't forget about the fact that it really does depend on what molecules cells have available to them. And remember, ketone bodies are only made in the liver when glucose levels drop and fat breakdown rises. The liver will then share the ketone bodies as well as any glucose made in gluconeogenesis with other cells so that they can then make energy. And it's the conversion of the glucose to pyruvate to oxaloacetate that allows the other cells like the brain to efficiently make ATP from acetyl-CoA that comes from the easy breakdown of these ketone bodies. Or if you're talking about other cells like muscle, it could be acetyl-CoA coming from those multiple rounds of beta oxidation. When you have too many ketones being made, either due to starvation or maybe a low-carb diet or in diabetic cases, you can enter into a condition called ketoacidosis, which occurs because the body can no longer buffer all the protons that are being made. Your body will try to compensate by using bicarbonate in the bloodstream to bind to the protons and produce carbonic acid, and this will then get broken down to water and a CO2 molecule. People that are experiencing acidosis might have a deep sighing breath where they're trying to get rid of all of the CO2 that's being made. There's also other physiological mechanisms that you'll learn about in your other courses that help bind to the protons or secrete them in the urine. But when your blood pH drops, usually below 7.35, this really can have huge consequences. So just take a second and think about what you've learned so far in this course. Can you think about why an abundance of protons can be bad biochemically? Remember, the protonation state of functional groups is key to how they interact. So too many protons can affect protein structure or function due to the protonation state of the amino acid side chains. But the same goes for all of the other macromolecules. pH really can affect their structure and or function. I mentioned that ketoacidosis can occur in diabetic cases, and this would be known as DKA or diabetic ketoacidosis. For the purpose of this course, we are going to look at the metabolism in the context of type 1 diabetes, where there is an autoimmune damage to the beta cells in the pancreas. And these are the cells that are responsible for sensing the influx of glucose and sending out insulin to help with the cellular intake of glucose for energy production. People with type 1 diabetes do need to take synthetic insulin injections to tell their cells to use any carbohydrates that are coming in from their diet, but they do have to balance how much they take along with their exercise regime to ensure that hyperglycemia and hypoglycemia don't arise, where hyperglycemia just means high blood glucose, either due to inadequate cellular uptake, and hypoglycemia is where insulin signaling doesn't get shut off and cells run out of glucose to make ATP. So let's look at the first scenario, or hyperglycemia. Hyperglycemia is bad in both type 1 and type 2 diabetes because that glucose molecule is quite reactive. When it reacts with other molecules, this is a non-enzymatic reaction, and it's called glycation. Now, don't get this confused with glycosylation, which we saw was a normal process for some membrane proteins. One of the molecules that can get easily glycated is hemoglobin. So when lots of glucose is found in your bloodstream for a prolonged period, it can enter red blood cells and react with the N-terminal valine residues found on the two beta chains of hemoglobin. The amine will form a covalent bond, or an amide bond, to the aldehyde and form a molecule that's called hemoglobin A1c. And this is actually a marker for high blood glucose. It doesn't affect the function of hemoglobin. This modification is far enough away from the heme binding sites, but we can detect the HbA1c levels in a lab to determine if someone isn't managing their diabetes correctly. When this reaction occurs with other molecules, however, it can damage them, and this can contribute to some of the other symptoms of diabetes. Now, these would be known as advanced glycation end products, or AGEs, and they can affect proteins in the bloodstream or maybe membrane receptors, and all lead to damage that we see either in the retina, kidneys, capillary, or neuronal cells. So let's just look at what happens when insulin is released from the pancreas or when someone with diabetes takes insulin. As your blood glucose rises, the presence of insulin inhibits the alpha cells of the pancreas, and these are the cells that are responsible for glucagon release. Insulin signaling to the cell will allow for the uptake of glucose, allowing for energy production and glycolysis, as well as the synthesis of fats, proteins, and glycogen using the energy in any excess carbons. Because the anabolic pathways are on, the catabolic pathways will be off, so we won't be breaking down glycogen, fat, or even synthesizing glucose in gluconeogenesis because it's only initiated by glucagon signaling. When your blood glucose drops, if you have too much insulin present, this will actually prevent the alpha cells from releasing glucagon. And remember, this hormone is supposed to signal to the liver to help restore blood glucose levels. Since insulin is still present, glucose entry and glycolysis will continue, and we won't be able to access any of the glucose in glycogen stores or make it in gluconeogenesis. And then hypoglycemia, or low blood glucose, will occur. So this can be very bad for your brain, since your brain is relying primarily on glucose for energy production. So this is why people with diabetes really need to monitor their blood glucose levels, say with a glucometer and a small pinprick of blood. And this will also allow them to measure and figure out how much insulin they need to take. When insulin isn't present, hyperglycemia can occur, leading to glycation of lots of macromolecules. The insulin injections will allow their cells to use the glucose, but also inhibits glucagon secretion. And if you're exercising and aren't eating, you don't want to have too much insulin present, because the liver won't be able to respond with glycogen breakdown or gluconeogenesis to maintain blood glucose levels, and this will result in hypoglycemia. Fat breakdown and ketogenesis can also produce acetyl-CoA for energy production, but remember, there are side effects for having too many ketone bodies made. If we look at how diabetic ketoacidosis arises, and this can happen if someone isn't taking their insulin, or maybe if they don't even know they have diabetes yet. In the absence of insulin, glucose can't get into the liver, or any other tissue for that matter, in order to make ATP. And fat breakdown will occur due to the hormonal response of this stressed, low energy state. The levels of oxaloacetate, or OAA, in the liver will drop, and the citric acid cycle is going to slow down, while the excess of acetyl-CoA from beta oxidation will lead to the production of ketone bodies in ketogenesis. If too many ketone bodies are made, ketoacidosis occurs, and this can lead to coma and death due to the insufficient ATP production, as well as the protonation of important proteins needed for cellular function. So it's really important that you understand the underlying biochemistry of type 1 diabetes, or really for any metabolic condition. My cousin's son tragically died at the age of 13 from type 1 diabetes because they didn't know he had it, and they didn't know the signs to look for. In addition to the fruity breath that we talked about, coming from the production of acetone and ketogenesis, that deep sighing exhalation may be an indication that someone is trying to get rid of excess CO2 coming from the blood buffering of acidosis. The 4 T's for type 1 diabetes are common symptoms to look for, so someone might be going to the toilet quite frequently, and this is known as polyuria, where they're trying to excrete and get rid of all the blood glucose, which would be known as glucoseuria, as well as ketone bodies and protons, which is really their kidney's way of excreting it, or known as renal excretion. When you are removing liquids in the form of urine, this is also going to deplete blood volume, leading to hypotension or low blood pressure, and you could pass out because of this. People that are exhibiting these symptoms might be quite dehydrated, since the presence of these molecules in the bloodstream will pull the water out due to osmosis, and the physiological response would be extreme thirst, which is known as polydipsia. They will be quite tired, either due to decreased ATP production, since they may not be using glucose as an energy source, and they might also be losing weight due to fat degradation. So do keep an eye out for the 4 T's, which may be signs of the onset of type 1 diabetes. Type 1 diabetes is a manageable condition. Even before the discovery and purification of insulin, low-calorie diets were one of the ways to survive, albeit not the best lifestyle. So once insulin is taken, this will allow for glucose to be taken up into the cells for ATP production, and polydipsia and polyuria will be eliminated due to a drop in the blood glucose and ketogenesis. This will allow the concentration of solutes in the blood to come back to normal, balancing out the water levels and dehydration. Remember, insulin is the anabolic hormone, so glycogen and fat breakdown will be inhibited. And if gluconeogenesis was adding to the amount of glucose in the bloodstream, it'll also be shut down. Any excess carbons that don't get used for energy could be stored as glycogen or fat. So just in summary, ketone bodies are made in the liver in all of our bodies when acetyl-CoA levels rise, so that other tissues can use them for energy. Type 1 diabetes arises due to a lack of the hormone insulin, and people with type 1 diabetes do need to take insulin and balance their injections with how many carbs they're consuming and their metabolic needs when it comes to ATP production and exercise. We can understand the symptoms of type 1 diabetes based on the biochemical molecules that are being made, and if you can understand what's going on at the cellular level, this can help you treat and manage the underlying condition. The last thing I want to leave you with is a thinker question. Even if you don't have type 1 diabetes, type 2 diabetes is on the rise, so we should all be thinking about maintaining healthy lifestyles to prevent extended periods of hyperglycemia, which can lead to the formation of those advanced glycation end products. What strategies are you now going to adopt to ensure that you do maintain healthy blood glucose levels? Do review this lecture before attempting the brain break questions on Quarkus.